what I'm going to do today is that we are talking about the adaptive and the innate defense. And what you will see that there are some slides, as I said, looks pretty intimidating. And again, you do not have to memorize each and every name of those. And just to tease you what I did, that almost for always, I may miss a couple of them, that you have these crazy abbreviations with the names. And just for your amusement, I just tried to put their almost all of their full name. And literally, you can ask probably a scientist who works in that field that what is the full name of some of these proteins, and many of them will not be able to answer because nobody just actually looks at it because they were just discovered by the scientist. And these names of these factors are just completely idiosyncratic. There is whatever they wanted and put in the paper, that sticks, that's it. So that's the reason that it is so difficult to keep track of all these signal transduction pathways due to all these crazy names. But that's not the goal. The goal is to just filter out the information that what is needed. Now, I'm not going to teach you immunology in this class. Remember that a biology 3050, which is a microbiology class, is a prereq for this class. And in that microbiology class, you have two separate chapters. You had innate immunity, and then you have adaptive immunity as a chapter. Remember that. Your adaptive immunity is also known as acquired immunity. And we are going to talk about some of the basics that's inevitable, but we will concentrate mainly on virus-related material in this chapter. And I have added a lot of extra material because with this COVID-19 situation, a lot of people are actually interested in this immunology part of it. And many of these things actually can enlighten you to understand that what is going on. So that's also another thing. And then we'll have some special topic chapters that will come up in some of the papers that the graduate students and the honor students are doing, that we will add those informations as well. All right, so let's start. So here, if I'm just following this book, which is the Principles of Virology, hold on one second. If I'm just following this book, they actually divided the immunity into actually three parts. I have divided into its innate and the acquired immunity, and they divided this into an intrinsic immunity as well, that you can see right here, which they put this anatomical and physical barrier along with that. And I did not do so. So I'm just talking about all of these as innate immunity. And then you have acquired immunity. And this is the norm that most of the microbiology or the immunology book actually do follow. And scientists agree to some extent and sometimes don't agree. And that's what it is. So let's first take a look at the overview of that. So just think about that these part of the immunity, also we just interchangeably use the word as a host defense mechanism, is just like a barriers or the walls. And they are constantly being attacked. We are just concentrating here on the viruses, but not just viruses, pathogens, like everything else. You have bacteria, protozoa, fungi, allergens, everything is attacking them. And then you have this natural anatomical and the barriers which will stop them, most of them. Some of them will go over, and then you have some intrinsic factors which will be in place to stop them. Then you have the innate and then acquired immunity. And sometimes if some of these pathogens get lucky enough that, and not always, but sometimes they do, can cross over that barrier, which is the adaptive or acquired immunity, then you may, again, make sure that you may see a disease, but not always. Remember that. Sometimes you may have this acquired immunity kick in and the disease could be silent. So not necessarily that a virus just comes over here and it will cause the disease. When this last barrier, acquired immunity, actually fails, the picture actually doesn't give you the exact portray that whole thing. If this acquired immunity completely fails, then only you actually see a manifestation of the disease. So that's basically what it means at the last stage. So uh, we divided this first part, that innate immunity, into tiers. So the tier 1, 2, and 3, and all these pathogens cross them in this fashion. So we are going to talk about these, and then we are going to take a look at this intrinsic part of the immunity, tier 2. So what is intrinsic? Intrinsic means that these are present already in the cell as a factor, or in the terms of a process. They're already in place. You don't need any kind of protein synthesis or signaling to start this. So you can easily see that the interferon is in here, and you will question that what do you mean by this? If I get a viral infection, usually the interferon is signaled. So why this is so? Remember that every day you are encountering some kind of pathogen, and there are some interferon in your system in a small amount. Now, what happens that if you have a real infection, this interferon amount, it goes up to synthesize more. And this is again a controversy that I just wrote down in your notes. So it could be really a blurry area. On the other hand, see this microRNAs, we talked about how it actually enhances or keep the genome of the hepatitis C virus protected. That's one of the viral strategies. But the microRNAs are silencing RNAs, which helps in some antiviral defense mechanism. And the CRISPRs that, like everybody in the streets even know, these are usually prokaryotic bacterial and archaic defense mechanism, which actually were developed for their infecting bacteriophage and other archaea related viruses. So the microRNA based and CRISPRs is not considered. This is a little bit, sometimes it's argued, but it's not so relevant in eukaryotic system, and it's not considered as the mainstream antiviral. That's why we are not going to talk about this. And I'm sure that what they are, you will study in some different courses. Then we are going to talk about this innate immunity. These are the cells and proteins which are present in your body, and they will rush to the site of infection if they are cells. And the complement system are a series, more than 30 different proteins which are present in your blood serum in an inactive form, and upon an infection that can actually get activated. If the innate immunity or the defense is not enough to stop the infection, they will call the adaptive immunity. So as soon as the innate immunity engages, it will start to release factors, which we call as a cytokines. And the cells, which are the T cells and the B cells, these are the lymphocyte, and they are present in your body in certain areas like a lymph node, spleen, and they will be called upon to take the measure, and usually most of the times are successful in clearing the infection. So that's an overview of that. And what I assume in here, that you have a basic idea about the immunology. That's the reason that sometimes I may not explain some of the things, and I added extensive notes in there that you can actually do a little bit of a recap from there. So let's start the material. So the first thing is that innate immunity, tier 1, that what we call as, these are the anatomical and physical as well as chemical barriers. So you have your mucus, saliva, stomach acid, which contain proteases. Then you have these bile salts. Remember, those bile salts are detergents, which can have some effects. Then tears, and your skin is a barrier as well. So again, very self-explanatory notes that I have given, but the mucus traps a lot of different organisms, and including the viruses, and it do contain, both the mucus and the saliva, they do contain secretory antibodies, which is called as an IgA, and they are already present, and these are non-specific with wide variety of binding domain. That means that they are not specific to a viral infection. Whatever comes in, randomly these secretory antibodies will try to bind to them and try to neutralize them. On the other hand, saliva do contain lysozyme, which degrade bacterial cell walls, the peptidoglycans, but not that relevant for the viruses. On the other hand, the stomach acid proteases, they can also interfere with the viruses. Bile salt contains the detergents, and remember that now you can tell that detergents are basically will dissolve any membranes. So that's one of the reason that most of the viruses which infect through the gut or the alimentary canal, they are not enveloped viruses. Enveloped viruses are cannot infect through the gut because they will be degraded pretty easily and disintegrated by these bile salts. On the other hand, say for example, poliovirus, rotavirus, norovirus. These are some of the viruses which actually can infect through the gut. These are none of them are they are just they have a very their capsid is just a protein shell. They are naked viruses, and that's why they can infect through the gut. And their protein shells are also well guarded against the protease digestion as well. That's one of the important things. And the tears also do contain these secretory antibodies, which is IgA, and the skin is just a physical barrier. So if the skin, what I mean by this is your surface of the skin, the dry skin, sometimes also like herpes viruses actually can go through the skin because you have the pores, but usually that's that's a good barrier as well. But any of your bodily opening, they are lined by live cells at the very top, but they're always lined by the mucus. So your mouth, nose, anal opening, urogenital opening, they're all protected by this mucus. So that's your first barrier, and most of the time that is good enough, but viruses can breach these as well. And one of the important thing is that you can see that if there is a cut in your skin, or sometimes just this arthropod or insect related vectors can inject the virus into your body. Say for example, Zika, West Nile, yellow fever, all of these are mosquito, dengue. These are all mosquito-borne viral infection, which is vectored by this arthropod and injected to your body as well. So this is your first part, the anatomical defense. Then we are looking at the next one and the overview that how this works, that the self receptor, we already know that as soon as that cell receptor is engaged by the virus, the cell actually knows that there is something going on. Now, not necessarily it will understand even at that point that it is a virus, not the natural ligand. So there will be some signaling going on, but you need some other signals to understand that there is a virus in the system. So, but the main thing is that no matter what, there will be signaling going on as soon as the virus is engaged on the receptor surface itself. So how we now recognize these foreign nucleic acids or the viruses which are binding on the cell surface or able to get into the cell. Now, again, a lot of information, but again, we are just going to filter the information that what we know. If you can, then you will see that it is not that complicated. So the first thing that what we are looking at, that there are some receptors, there are 10 of them in the human beings, 12 of them in the mice. These are called the TLRs. These TLRs are the toll-like receptor first discovered in Drosophila, and they are development related in the dorsoventral axis. But they were, and these TLRs or the toll-like receptors are on the cell surface. They could also be inside the endosome, and these detect foreign signatures when they are found. And there is a word which we use is PAMP or MAMP. What we mean by that is a pathogen associated molecular pattern, PAMP, or sometimes we call it is a MAMP, microbe associated molecular pattern. Say, for example, for a bacteria, peptidoglycan is one of those PAMP, which is never found anywhere in any of the eukaryotic system. So immediately that is a PAMP or pathogen associated molecular pattern. And the same way, the double-stranded RNA and some of these RNAs that the CPG motifs, these are also specific for the viruses because these are not found in a normal cell, and these are considered as molecular patterns, and these are basically understood by these receptors. So what are those? Say, for example, there could be TLRs on the cell surface that can bind to the viruses. Also, it can bind to its natural receptor. And then once they're internalized, this double-stranded RNA, you already know the PKR can actually sense that, which will block the translation or try to block by the phosphorylating the EIF2 alpha. That's a global block of translation, and you already know that the viruses have processes just to bypass that. Another double-stranded RNA is sensed by this RIG1. That's also another one. This is in the cytosol. On the other hand, the endosome, if the viruses are going in, there are several TLR 3, 7, 8, 9. Out of that 3, 7, and 8, they can actually understand the RNAs, double-stranded, single-stranded, including silencing RNAs. On the other hand, this CPG motifs, that means you have a cytosine phosphodiester bond, and then you have a guanosine. This is basically CGs on a piece of DNA. And these particular motifs are understood by these TLR9s. And each one of them they are transmembrane proteins, and they do have this MID88 or TRIF. These are some of the cytosolic part, but these are the ones which are activated. And ultimately, no matter what, with all other different processes, what ultimately happens that if you just look at that, no matter where it comes in, they will all activate the NF kappa B, that is one of the nuclear factor kappa B, or this IRF3 or IRF7. These are the interferon regulatory factors, IRF3, IRF7. What we mean by this, your IRF3, NF kappa B, IRF7, all of them are transcription factor. So once they bind to that, you will have a rapid gene expression, and the cell will produce interferons to make them in an antiviral state. It will also produce these inflammatory cytokines like tumor necrosis factor alpha and interleukin 6, interleukin 12. There are more than 30 of them, depending upon the situation. And the interesting part is that once these are produced, these can produce more than 1,000 plus gene expression. And you can actually look at these that how many genes are activated by this NF kappa B. It is kind of mind-boggling, and the functioning of that in the some of them are not even known today itself. So this is the sensing of that what happens in here, and RNA is detected by the RIG1. On the other hand, this MDA5, TLR7, 8. So these are the TLR 3, 7, and 8. So that's one of the things. On the other hand, DNA is detected by the TLR9 and another, which is called the C gas. And the C gas is the cGMP synthase, which synthesize this cGMP bound by 2' and 5'. So you have a, so what do we mean by the cGMP? So it is a cyclic GAMP. That means a guanosine unbound with an AMP, adenosine monophosphate, by a 2' 5' linkage. And this is a signaling molecule. So this signaling molecule binds to a protein, which is a transmembrane protein called STING. Through the ER, it is trafficked through the to the Golgi. You can see that it is still at the Golgi stack, through the Golgi stack, but this is the cis face of the Golgi, where it activates the I kappa factors, which will essentially take the signal to the I kappa B, which is bound to the NF kappa B. NF kappa B is released from this condition in the inactive form. That will cause the gene expression. Also, through the TBK, this interferon regulatory factors will go in, and it will stimulate gene expression. So these are the antiviral measure for the DNA and the RNAs. So let's take a look at that, how the virus actually escaped from that. And you can easily see right here, say for example, Epstein-Barr virus actually do something here. This TRAF, you can see that is ubiquitinated, this NEMO regulatory subunit of this I kappa alpha beta, and also the I kappa beta, which is bound to the NF kappa B, in that to make this inactive, this I kappa B must be ubiquitinated and degraded so that this NF kappa B, which is the transcription factor, is released and translocates into the nucleus. So this BPLF is the protein found in this Epstein-Barr virus, which blocks this all these pathways. So the thing is that it needs to be degraded so that the next signal can be can form. This needs to be degraded for the next signal to be formed. And I kappa B needs to be degraded by this ubiquitin annihilation before this part is released and goes on. So all these are basically blocked. So basically BPLF is a deubiquitinase which doesn't allow the deubiquitination and blocks these pathway. There is also another protein which is known as this A20 that also blocks directly the TRAF6. Easy, you can see that. So these are the ones which are important in the sense that the Epstein-Barr virus is a herpes virus, and that's one of the reason that this is actually in place, because they can form a long-term they can reside in the cells for a long time, escaping from the cellular immunity. And also this measles virus, which is a paramyxovirus family, produce a protein which is called A20 that can also hamper or inhibit this TRAF6 signaling. All right, so after that, we are looking at the interferon we were talking about and what interferon actually does. Interferon is a defined boundary that when you have a cell infected by a virus, it will start releasing these, producing these interferons. And once these interferons are produced, that will disperse through the system and it will make like a fire divide. And the infected tissue is already in there, and the unaffected tissue under interferon will go into a state which you call as the antiviral state. And that is done by the interferon. So that's the basic concept. And they are released from the virus infected cells and uninfected sentinel cells. And these sentinel cells are usually the macrophages, and sometimes they are the neutrophil as well, but mainly the macrophages and the dendritic cells. So these are some of the cells in the tissues that's in there releasing this cytokines. So now we are looking at that how the interferon actually works. And again, just an overview of that. There are three types of interferon: alpha, beta, and then you have gamma, delta interferon as well. And these interferon receptors, you can see that these are hetero, sometimes they are dimers, but heterodimers. Sometimes they are they are tetramers, and they come in all shapes and sizes. So say, for example, for the type 1 interferon alphas, many different types. There is one which is called an IFNAR1 and IFNAR2. These are the exoplasmic surface of these receptors, and so on for the other one as well. Usually, they have a transmembrane, not usually, always they will have a transmembrane domain, and the cytosolic part is associated with this JAK or the TIKE. So this JAK is a Janus-associated kinase, which is basically a phosphorylating agent. And then the TIKE is a tyrosine-like kinase. But again, remember that we talked about, or you have learned somewhere else, that there is a receptor which is called these RTKs or RPTK, receptor tyrosine kinase. Receptor tyrosine kinase has a receptor on the cell surface, but these TIKEs does have a domain like tyrosine kinase in the cytosol, but the receptor part is something different. It's not a receptor tyrosine kinase. But phosphorylation is the main thing. So what these things actually does, that when they are bound with a ligand, all of these will stimulate a pathway through this STAT. And this is basically, the STAT is the one of the most important part, which is a signal transducer, basically. These signal transducer get phosphorylated with either JAK1 and the TIKE1, and through this intermediate, these signal transducer will form this ISGF2, and these are the interferon stimulatory regulatory element on the DNA, And this can make a large, more than 1,000 different types of genes can be activated. And you can just look at this and take a look at that, how many of these are actually produced in the cells due to the interferon. And the Janus, how it was a name, and it was kind of funny that I'm just telling a story that when it was these Jack kinases were discovered, the scientists told us name is as a Jack. First, they told it's just another kinase, uh, just another kinase 1, 2. They named it that way. But when they tried to publish the paper, they thought that that may be too kind of like a non-scientific or may not be that serious, and they actually named it the Janus kinase with these two faces. So they just face two sides like this. You can easily see two sides of that, and that's the reason that the name is as a Janus kinase. All right. So after that, we are looking at the big picture of the interferon signaling, and you do not have to remember any of these intimidating signal transduction pathway. But the overview is that just to understand that your RNA virus, DNA virus, viral glycolipid, these are all detected, and all these intermediates finally, or on the other hand, you can see that we talked about this before, the TLRs detect all these. So no matter what, take home message, viruses outside the cell, as soon as they deliver their nucleic acid, they are detected by the RIG1 or the MDA5. The DNA is by the C-gas. We have seen that. And these TLRs can also detect this. And then with the signal transduction pathway, they finally produce these interferon stimulated genes. Once this interferon alpha and beta is produced, these are both type 1 interferon, they are secreted outside this cell, which is infected. That is the important part. This is the cell that is infected. They are releasing these interferon, and the other cell have the interferon receptors will receive this. Bless you. So other cell will receive this. Once they receive this, there will be this signaling, which will be this part right here. That signal goes on, and this IFN stimulated genes, which you talked about, there are thousands in there, will make these cells or convert these cells into an antiviral state that we talked about. So that's basically the idea. And just two of the examples out of those thousands plus genes that we talked about, just to have an idea. One of them is a PKR, protein kinase R. And RNase L is, we talked about that. RNase L is also a viral RNA degrading enzyme. These are actually produced much more in number in that in this antiviral state. So that's basically how the interferon signaling works in a viral infection. So the viruses actually do have countermeasure, and you can easily see that this is completely self-explanatory, that they can interrupt with the cytokine production. They can interfere with the cytokine action, encode homologs of cytokines to block receptor, encode soluble cytokine receptor to neutralize cytokines. These are kind of a decoy mechanism. On the other hand, they may alter the cytokine signaling pathways as well. There are many different types of molecules. Just have an overview of that. There are too many to list, but that's how the viruses always can countermeasure these measures of your innate immunity as well. So the next type is the intrinsic part, that what you have. And we are not going to talk about that in detail because you know this already what happens. We're going to just touch that. There are some other measures as well. One of them is autophagy. Autophagy is basically for the cell recycling of the cell organelles, but that can be used to kill the viruses as well. But unfortunately, this process is used by some of these viruses for their replication. Say, for example, poliovirus or coronavirus, they actually co-opt to use this as a replication intermediate and also for the dissemination of the viruses. So they hijack this machinery. Apoptosis is a process, although I say apoptosis, but technically, the correct pronunciation of that should be apoptosis. It is not, you do not, the P is silent where the word was actually derived from the Greek letter. You should not, the second P should be silent. So that's like how you do this pterodactyl, like it starts with a P, but it is silent. The same way receipt, the P is silent. So the same way it should be pronounced in apoptosis, but it's a habit. Sometimes I may remember, but sometimes I can say apoptosis, and I excuse me for that. But the apoptosis is the process by which the cell can fragment itself in a condition when it is need to die, particularly in a viral infection, and that can help to spread the viral infection. But unfortunately, what happens, you already know that this process of apoptosis is blocked by many viruses, whereas some of them can also use them to their at the late stage to kill the cell and get out of the cell. So we have already saw that both of these processes can be proviral or antiviral in the effect, but they are in place mainly for the antiviral effect, which is modulated by the virus. So here are some other examples of the intrinsic immunity, and this is a classic example, very specific. There is a protein which is known as ApoBec3, apolipoprotein B, and this protein is a very interesting one. What it does, it is a deamination protein. So if there is a viral RNA that's going on, particularly in HIV, if you just look at this area, what it actually does, that if you see that this is the viral RNA, and this is a 5' to 3' plus strand RNA, and you have a sequence like GG and the GA. So first, by reverse transcription, that which will produce the minus strand DNA. So GG, you got CC, and then you got complementary GA to CT. So this protein will induce some mutations, which we call as a deamination of these cytosins. And now you can see that it introduces another U instead of this C, introduces an U instead of C. One C is left in there. So I mean, like, it will try to deaminate all the cytosins, but it may not be, but just two of them. But see that downstream what happens. You have this mutation in these two places. Now, when you synthesize the plus strand, you got an UA and UA. And then you will synthesize again the double strand, the minus strand, or the 3', 5' strand of the double strand, you got an U and U. So you can see that just this deamination step introduce a large number of mutation in the reverse transcription process, and this will destroy the DNA completely, and it will be unusable. So this is an intrinsic mechanism for the retroviral defense that people think that evolved many, many years ago for the retroviral infection. Now, HIV can counteract that by a protein which is known as VIP, VIF. So this VIP binds to this ApoBac3G, and this is the overall complex. And then it basically adds the ubiquitin residues onto the ApoBac3G, and it's degraded in the proteasome. So what happens that you can easily see this ApoBac3G is a protein which can degrade these proteins, but it cannot, because this VIP is already in this. So the VIP is produced and packaged into the virus to a certain extent, a small amount, and that is good enough to inhibit the function of ApoBac3G. So you have normal reverse transcription without mutation due to this VIP. If you remove the VIP, you just make a recombinant virus where you don't have the VIP, you can see that you can enhance the degradation of these RNA. So this is an age-old mechanism, an intrinsic mechanism to probably stop the retroviral infection, very specific. So another way that the silencing which is known as the epigenetic silencing, also can stop the viral DNA replication. And sometimes it is used by many of these herpes viruses to their advantage, and but they can actually escape from this process as well. And this is just done by the acetylation and deacetylation of these histones, basically. And you already know that when the DNA is compact like this, the genes are silenced and the cell can actually do that. When you have these acetylated histone tails so this is done by the HDACs or histone deacetylases, whereas the histone acetylases acetylate the histone and they become loose. And at this point, you can have the gene expression. So, say for an example, if you just look at these example, human cytomegalovirus, human herpes simplex virus, EBV, Epstein-Barr virus, all of these, except this adenovirus, all of these are DNA viruses, and these are supposed to be silenced by this process by the cell, and this is done in some specific site, which are called the PML bodies. So when you see that there is no interferon, you don't see these PML bodies, or much less number. But when you have the interferon, these PML bodies are actually the sites where it is thought that this silencing of these viral genes goes on. But there are measures of the viral, like say, for an example, HCMV-PP71 is a protein which degrades the DAX, which is a histone deacetylation of the protein, and help in the viral replication, or help in the viral transcription process by opening up the DNA. And the same way that other proteins can do this as well. So that's another measure that epigenetic silencing is supposed to suppress the viral DNA replication, not the DNA replication, viral DNA transcription, but there are measures to counteract that as well. So after that, we are looking at these natural killer cells, and natural killer cells are also the players in the innate immunity. And what they do, that they can sense that whether a normal cell is a missing self or altered self. What we mean by that, that if somehow a normal cell in your body is altered some way or doesn't have something, it can understand that, and then it will going to kill that cell along with the virus. And there is an inhibitory receptor, and along with that, you need an additional activating receptor, and both of them are required for the killing purpose. Otherwise, sometimes if you just have just one layer of security, sometimes it can kill a healthy cell, as if it sound like it could be a false positive, and it could be detrimental to your body. So what you are seeing right here, that this is a target cell and this is the NK cell, and just have to have an idea that what is needed. So this MHC class 1 homolog, this is expressed in the cell in a plenty of amount, and this MHC class 1 receptor is of the NK cell can recognize that. And when it recognizes that, it understands that everything is going on well in the cell. Now, understand this very carefully. Upon some of the viral infection, what happens, sometimes this MHC class 1 homolog, if their amount, on the other hand, is reduced, it will produce some of these HLA-E, and that is also recognized by MHC class 1. But what happens, that this HCMV actually synthesizes this HLA-E, escaping the potential of NK cell recognition. So this is basically a decoy mechanism that is actually used by this HCMV and to confuse the NK cells. On the other hand, you can see that this is an activating receptor that is absolutely required for the NK cells in addition to these inhibitory receptors, but this is also blocked by a number of other receptors as well. And you also see that these are some of the other stimulatory material, like a cytokine receptor, also recruit the cells at the site. And also there are other inhibitory receptors which can directly bind to some of these viruses as well. And these can be used to even infect the NK cells. So this is basically the process by which virus infected cells are killed by the natural killer cells, which are present as your innate immune defense, but there are viral countermeasures as well in place. So the next one is a complement system. And what complement system is a complement system is a number, more than 30 different proteins which are present in the blood, and each one of them is activated by a different pathway. One is a classical pathway. When antibody is bound to a microbe, mainly bacteria, it starts a cascade. On the other hand, there is a lectin pathway where the mannans or the mannose are bound, or the carbohydrates are bound. They actually trigger those. These are some of the carbohydrates found in the microbes. And then there is also another alternative pathway when one of these serum proteins C3b is coats the microbes. But this pathway is more of the function in bacterial defense. But nonetheless, they can be implicated in some of the viral infection as well, because of some of the specific things. Say, for an example, one of the triggers of this is an antiviral response in the process of inflammation. So this complement system can do this inflammation, which is basically alerting your immune system that something is going on. So on the other hand, there is another process that if the virus is coated by this particular protein, which is called the C3b, there is phagocytosis that can take up this virus coated with the C3b. This is called opsonization. So again, repeating, C3b coated virus can be phagocytosed by the phagocytic cells like your macrophage, and that process is known as opsonization. Another particular process is by this whole cascade, this system produce a membrane attack complex. This membrane attack complex is made up of many proteins, and usually in a bacteria, they are inserted into the bacterial membrane, like the phospholipid bilayer, and they will make pores and perforate the membranes, and that's the process of the killing. The same process can happen only to the enveloped virus, and you know the envelope is basically a phospholipid bilayer, which is derived from some kind of cellular membrane while the virus buds out, either ER, Golgi, or plasma membrane, or even nucleus. But it's a cellular membrane with phospholipid bilayer, and these membrane attack complexes can be inserted into them to make them, and they will not be able to mature without that membrane. But again, some of the viruses like alpha virus glycoprotein C and this pox virus, which is a variola, this is a spice, is a smallpox inhibitor of complement enzymes. And all of these can also countermeasure the complement system as well. So the next is that we are looking at that there are some specific cells of the innate immunity, which we call as a professional antigen presenting cells. So the function for them is to capture these virus or other microbe antigens and display onto their surface and present into the cell, particularly of the adaptive or acquired immunity, which is a T cell and a B cell, for the next step. So what are these? One of these are called the dendritic cells. They are present underneath the skin. They are constantly moving around in every tissue looking for pathogens. Macrophages circulate in the blood for a short time as monocytes. And these monocytes leave the blood and then mature in the tissue as macrophages. And these are your phagocytic cell as well. And you can see the detail. And the B cell is not actually a part of the innate immunity. B cell is the part of the acquired or the adaptive immunity, but it can also present antigen to its own system. And that's the reason that I just put it in there. But remember that this is not the part of innate immunity, but B cells are the part of the acquired immunity, which we're going to talk the next. On the other hand, you can see that there are always processes by which these cells are infected as well. Say, for an example, that HIV can infect these antigen-presenting dendritic cells. And there could be two types of infection, a cis infection or a trans infection. see that what is happening in the cis infection, that it just binds to the receptor, and then these cells are infected. Once these cells are infected, they actually do kill, and the viruses are released to infect more cell. On the other hand, this is a trans infection. What happens, that this is the interesting part, these viruses are put onto right here, on, onto a particular C-type lectins, which is known as this DC sign. It's a huge, big name of that. I put it in the note. You can just look at that what is DC sign is. A very funny, complicated name. But this is just a C-type lectin onto which the virus can be put on into the endosomal vesicle. And that can be transported onto the membrane. And then from here, it can infect a CD4 T cells, which is known as a helper T cell. And this helper T cell is one of the final target of the HIV, which actually destroying these CD4 T cells or the helper T cells actually suppress the acquired or the adaptive immunity, which we are going to see in a second. So infection of the DC by itself and the virus dissemination is a cis infection, same cell. When it goes from the DC to the CD4, that is called a trans infection. So that's basically the two differences in between. So the neutrophil is not that implicated into the viral infection, mainly for the bacteria, but still they can do something. For example, they can make some proteins like alpha defensins, which can make pores into the membrane. Also, they can produce this oxidative ROS, or the reactive oxygen species, which are the superoxide, O minus is superoxide, which can also have the antiviral activity. Also, they can also make these long nets, which are the neutrophil extracellular traps. And these long nets are basically, which are made by these DNA, by making these DNA nets, basically. And they can also trap some of the viruses as well. So these are some of the overview of the neutrophils, but this is not the main antiviral mechanism, is not shown by the neutrophil compared to the other things. So the next, what we got, that this is the lesson learned from the immunity, is that there are some classic inflammatory response that happens due to this innate immunity, which is the heat, swelling, redness, pain. These are this. And due to this inflammatory response, the adaptive response is basically triggered due to this inflammation. If there is no inflammatory response, the adaptive response, which is the next, will not be stimulated. And this is the basis for the vaccine. So sometimes you just know that the vaccines actually do contain an adjuvant, and there could be many different ones. We're going to talk about that, which actually cause the pain in the site. And this is the inflammation, which actually make the adaptive immune system alert that something is going on, and that increase the efficacy of the vaccine. We're going to talk about that in a separate chapter. But this is the important one, all the viruses must encode at least one regulator of the intrinsic or innate defense. Without that, they will not be able to cause any disease or cross the barrier. And these could be all of these, you can see that sensing, IFN production, signal transduction, somewhere it has to make at least one regulator of the innate defense. So this slide, I'm not going to talk about that. But again, if you have not taken immunology, I highly suggest, I wrote that this is an important slide for the understanding of the innate immunity and how it is connected to the adaptive immunity. And there is nothing new information in here in terms of the viruses, but this is basically a refresher slide for your background. So I will just leave this to you guys, and definitely I have just put in the notes that this is a very important summary slide, and the following note is critical for the conceptual comprehension for that. So just make sure you read this very carefully, and you will be able to connect the innate and the adaptive immunity if you do so. So now what we have seen, that just in a nutshell, that the virus infects the cell, it releases these cytokines. These are small chemical molecules which will basically alert these dendritic cells, which are initially immature. Now it becomes mature dendritic cells, and it will carry some information, like here, right? You are seeing that this is a viral antigenic peptide. It will carry this to the closed lymph nodes and present this to the T cells. Now, here, the T cells and the B cells were sleeping. They will come back to life, means that from the naive state, which they are, they will be activated and will become effector T cells and the B cells. The B cells will secrete the antibody, whereas the T cells will produce this cytotoxic T lymphocyte, and these will do this direct cell killing, or the virus infected cell will be killed by this process. And usually this clears the infection. So that's the connection. So let's take a look at how these T cell activation is done by the dendritic cells. So here is an immature dendritic cells, and what happens that when the viruses are bound in them, they will internalize these viruses, and they will digest those virus proteins, and they will load these viruses onto this particular MHC class 2s, which are known as the major histocompatibility class 2. And basically, this is a presentation scaffold. So these particular proteins will be cycled back onto the cell surface along with this viral antigen sandwiched in between their chains. Now, this is a mature dendritic cells, which is displaying these viral antigens. Now, this will be presented to a naive T cell, and it will understand that there is a signal right here, which is basically a virus, which is foreign. Now, this naive T cell will become activated T cells, and it will release a number of cytokines, and you're going to see that will activate the adaptive immune system, which is the next part. So what are the effectors of the adaptive response? So you can easily see that these are all the antigen, like including the viruses, and there are two arms of this process. One is called a humoral response. One is called a cell-mediated response. So the cell-mediated response happens first. So how it happens step by step. So right here, you can see that there is in the bone marrow, these T cells are produced, which mature in thymus. And now these actually stay after maturing in the thymus. This is production. Then this is maturing in thymus. Then they will stay in the lymph node in a naive condition. That means that they are just sleeping in there. Upon infection, upon an antigen presentation by these dendritic cells, they will become activated. And there are two things that will happen. There will be a burst of these cytokines right here, which is that T helper cells will do that. And another burst of cytokine, which will, from the T helper cells 1, that will activate the CTL precursor, which is also a T helper cell. So there are just only two types. One is a CD4 T cells, which we call as a T helper cells. Activation is the function for that. There's another lineage, which is called the cytotoxic T lymphocyte, and these are responsible for direct cell killing. These are called the CD8 T cells. So they have these CD4 receptors here, and these have the CD8 receptors. So CD8 is cytotoxic T cell, CD4 is helper T cells. So these helper T cells, upon activation, will activate these CTL precursor, and now this activated cytotoxic T cell will direct cell killing. This is known as the cell-mediated response. This is one arm. On the other hand, this T lymphocytes, helper cells, when they put all these cytokines or the signaling molecules are released, they will activate the B cells, and these B cells were also in the also sleeping in the lymph node. And although these are activated, but the virus particles can directly bind to these antigens on the B cells as well, both of these processes. And then these B cells will change. So an inactivated B cell actually do have these antibodies on its cell surface. So you can see that the antibodies are on the cell surface bound to the plasma membrane, which can also detect the viruses. Once it detects these viruses and activated by these cytokines, this changes to a plasma cell. It no longer actually produce these membrane-bound antibodies, but they will change the isotype of the antibodies to secreting antibodies. And they can just secrete about 2,000 molecules of these antibodies per second out of a plasma cell. Huge number of antibodies are produced in these cells, and that is known as your humoral response with the B cells and secreting antibodies. And this is the process by which the T cells and B cells actually interact through these MHCs, where you can easily see that how the T cell is presenting these MHC related antigens to the B cells. So that's basically the overall process that we can see. So the next, what we are going to see, that the viruses can actually modulate the function of the T helper cells and the T helper cell 1 and T helper cell 2. So if you just look at this previous slide, here there is a burst of a Th2, T helper 2 cytokine. And here you can see that there is a burst of T helper 1 cytokine. Both have different functions. T helper 1 activate the cytotoxic killing. T helper 2 cytokines activate the B cells. So here what you are seeing, that when this cell is you can see that detected, this IL-12 is secreted from the cell. This is supposed to activate the CD4 T cells, and this pathway activate the Th1 response. And this is the appropriate at the initial stage, because without activating these T helper cells, you cannot activate the immune system. So that's one of the important parts. So this needs to be activated first. On the other hand, at the later part, the Th2 response is actually needed later, which will favor the antibody production, extracellular pathogens such as parasite, virus particles, which is this. First, it needs to do this, then it needs to do this. So there is a balance in there, and this balance is skewed by these antiviral responses. So say, for example, EBV, Epstein-Barr virus, produce an IL-10 homolog, which is an antagonist of this IL-12. And also measles virus elevate this IL-10. IL-10 is an antagonist of the IL-10. One produces homolog, another elevates that, which is the modulation of your own IL-10 or interleukin-10. So what happens that this pathway, which needed in the initial activation against the virus, is blocked, which is the antagonist of the IL-12. And the virus promotes right here this pathway. That's the reason that T helper is skewed, and that actually causes the antibodies to release to activate the B cells to release the antibodies, but that is not actually needed. So the initial damage is done by skewing is this Th1 and Th2 response by these viruses. Here we are looking at that how the T cells are activated. And here you need to remember some of these numbers. The CD4 T cells, which are also known as a T helper cells, contain a CD4. And this can recognize the MHC class 2 molecules right here. On the other hand, the CD8 are the cytotoxic T cells, which killing is the function, express CD8, and they can only recognize MHC class 1 molecules. And along with that, they have also other accessory receptor that like CD2 or 28 or LFA1. These are also needed for the recognition, but this is the main important part. So where you have this MHC2, where you have MHC1, and what does that do? So MHC class 2 is present in an antigen presenting cell. When this antigen presenting cell is presenting a viral peptide being displayed, T helper cell is activated and producing these T helper Th1 cytokines, Th2 cytokines. On the other hand, all nucleated cells do have these MHC class 1. When the cell is infected by the virus, it shows the viral peptides through an MHC class 1. That means this is an indication I am infected. This cell cannot go anywhere. The only fate of this cell is that killing of this cell, which will stop the viral replication further. And this is recognized by the CD8 to kill the cell. Here, this cell is not infected. This is antigen presenting cell. It is informing the T cell. Here, it is presenting that I am infected, need to be killed. So two different functions. And we are going to see that how these are actually presented to the T cells by these processes. So the first one is the endogenous antigen presentation by this MHC class II. It is produced in all nucleated cell. So here what we are seeing that you have an infected cell, and no matter, even in the infected cell, the cell is able to capture some of the viral proteins and degrade in the proteasome and make these peptides. These are basically the remnants or the fragments of the viral peptides, which go into the ER through these TAP1, TAP2 transporters, translogon-like material. Once they actually go into the ER, they are loaded onto this MHC class 1. And again, there are alpha chains and the beta chains, heavy chain and the light chains. Those details are not necessary. But once they are loaded onto this MHC class 1, they actually are trafficked the same way. This is the secretory pathway from the ER through the Golgi. They are trafficked onto the cell surface. Once they are trafficked to the cell surface, CD8 can recognize MHC2. Remember that 8 and I'm sorry, the MHC class 1. Remember that is 8 and 1. So you can remember that number 81 is the 8 and 1. So the cytotoxic T lymphocyte, when an endogenous antigen is presented, will kill this cell. So that's basically happens when a cell is infected. On the other hand, you can see that there are a number of measures where the MHC class 1 is countered by these. And you can see that lentivirus is a retrovirus VPU, HIV NEF, these are the retroviruses which actually affects this MHC1 synthesis or dislocation to cytoplasm. Rest all are, if you just look at these viruses, all of these are herpes viruses, and except this adenovirus, all of them are herpes viruses as well. So you just only have one adenovirus and two of these retroviruses. And they have all of these different functions. So if a question could be that what kind of question usually is asked from this slide? Do you need to remember all of these and corresponding function? No. Usually most of the common type of question is that which of the following is a process by which viruses can counter MHC class 1, and usually these are given, and most of the time these are all of the other. But if you just really see that somehow there is a question like this, which of the following is not a process by which these are countered, and suppose you find a question which is say like this, say which will block the complement pathway, completely out of the blue thing, which is not related to any of these things. As soon as you can identify that MHC has nothing to do with the complement system and can distinguish that is not the correct answer. So that type of question. So just overview of that. Not individual things are not necessary. So after that, what we're looking at, that the exogenous antigen presentation, on the other hand, is done by the MHC class 2 by the antigen presenting cells, and some other cells upon interferon gamma exposure can also do that. But here the cells are not infected. This protein or the viral protein or the virus is taken up by the endocytosis or the phagocytosis by, say, a macrophage or a dendritic cell, degraded into the endosome, late endosome, and then loaded onto an MHC class 2. MHC class 2 is very clever in that sense that it is blocked by an invariant chain so that it cannot load any of these its own proteins. And when it is trafficked into this low pH fusion vesicle, you can see that the viral proteins are actually degraded and it is loaded onto this MHC class 2, trafficked onto the cell surface, and see that CD4 or the T helper cell recognize this MHC class 2 and that will trigger all the downstream event of triggering the whole of the adaptive immune system. And some of the interesting thing that these are very diverse genes, and no matter what, remember that your MHC class 1 or class 2, they can make this groove any combination so that they can literally hold millions of different combination of peptides to present them. And this is done by the combination of these many of these genes. And in an inbred population, the diversity of these MHCs actually go down, risking them in immunocompromised conditions. Now we are looking at these antibody neutralizing antibodies, and I'm not describing the antibody structure. These are from somewhere else, and you can just read it just completely by itself. But the neutralizing antibodies can bind to many of the steps in the viruses. In the virus, it can bind to the virus itself so that it cannot bind to the receptor. It can bind to many of these processes. We are going to look at that. Now, there are many types. There are 5 isotypes of antibodies. One is called an IgM. These are the 5 types of antibody, and you can just look it up by your own. And these are produced at the initial stage. And there is also an IgA. This is a double antibody, 2 of them. So this is one antibody molecule. IgA will have another one on the other end, whereas the IgMs are the pentamers. These are non-specific antibodies. That means they are produced by the cells in a very large amount, in very, very large amount. And what it actually does, that it will circulate throughout your system and will try to catch the viruses. But these isotypes will be changed to the IgG, which is very specific to the specific virus after your infection, which will produce the neutralizing antibody. Important for recovery for infection. Read the notes. The important part is that even the President Trump was given that trial medication of this Regeneron antibody cocktail. That was a mixture of some of the monoclonal antibodies that we are going to discuss in a special topic. So here is a process that how these IgAs are secreted, and this is a transcytosis. So these B cells secrete this IgA, and I said that these are these double antibodies. You can see that one antibody, I said that it is another one is attached with this secretory piece. And by this transcytosis from this basal surface, they are transported onto the apical surface, and they can be on the mucosal lining. They are present in your gastrointestinal tract from your mouth to all the way to the anal opening, looking for viruses as well. So these are secretory antibodies on the mucous surface. Here we're looking at some of the neutralizing antibody, and this is a rhinovirus 14. And just what are the processes that can be done? You can see this 1, 2, 3, 4, 5. They are very self-explanatory and just shows that how these antibodies can block the viral infection. But why we cannot contain the cold? There are antigenic drift or small mutations, and each time there could be many different combination of viruses which can cause cold, and that's the reason that we have this recurring thing. Here's a very interesting idea. Just the last 2 slides of that. This is the spike. This is the hemagglutinin of your influenza virus. And you can see that there are many different ones. And this one, these 1918 H1N1 actually appeared back again in 2009. This was a pandemic in decades. Why this happens? Because you know that it's a segmented virus and it can change its fragments like the hemagglutinin or the neuraminidase. And this is basically the which is called the genetic reassortment. Due to this, they can just come up with all these combinations and fool the system. So let's take a look at what happened here. So this is a paper where you have this HA bound to an antibody right here. And this structure was solved and came out in the Science paper after that pandemic. And this antibody was taken from a person in South Carolina who actually is a survivor from the 1918 Spanish flu. So just imagine that this person was alive in 2009, and he did not get the infection of these H1N1 virus itself, but his body produced these antibodies, which was isolated, bound to these viral, the HA protein, and the crystal structure was actually solved. So here is all the different types of strains of the hemagglutinins. And when it was titered with this antibody, you can see that this was basically the H1, South Carolina 1918. You can see that this is how it is declined, whereas it did not do with the other one as well. So this actually shows basically that it can last a lifetime. And this is basically an eerie picture that the host subsequently infected by the same virus, the response will be rapid and specific. And that's the reason that the adaptive immunity provides the memory. Innate response don't have a memory. And that's the difference between the main difference between the adaptive. So there are B cells and the T cells. They can live a lifelong in your lymph node and can be called upon immediately when you see the viral challenge at a later time. And that is the memory for vaccination. But no matter what, still all of them can be breached and we can get a